Therapeutics. Welcome to Coya Therapeutics 2026 ALS Education Webinar. I'm Dr. Fred Grossman, President and Chief Medical Officer of Coya Therapeutics, and I will be moderating the webinar. Coya is dedicated to developing combination immune therapies for neurodegenerative disorders, including ALS and FTD. We currently have an ongoing clinical trial across the United States and Canada of COYA 302, which is a combination immune therapy for ALS to potentially halt or slow progression. We will discuss this in more detail later on in the program, but if you or your family member have been diagnosed with ALS and you have an interest in participating in the clinical trial, you can go to trials.coyatherapeutics.com for information and to view the participating sites in this ongoing trial. I would now like to introduce our distinguished panel of ALS experts, Dr. Stanley Appel and Dr. James Berry. Dr. Stanley Appel is the Distinguished Endowed Chair for the Treatment and Research of ALS, co-director of the Houston Methodist Neurologic Institute, and chair of the Stanley H. Appel Department of Neurology of Houston Methodist Hospital. Dr. James Berry is Winthrop Family Scholar in ALS Sciences, Averill Healy Endowed Chair in ALS, and Chief, Division of ALS and Motor Neuron Diseases of Massachusetts General Hospital. Welcome to you both. Today we will discuss the ALS patient experience, the therapeutic landscape, mechanistic insights, the rationale for Coya's combination of low-dose IL-2 and CTLA-4, and an overview of the ongoing Coya All-Stars clinical trial in ALS. Following the discussion, we'll have a brief question and answer period. So let's start with you, Dr. Berry. From your perspective, what is the biggest unmet need for patients living with ALS today?
Yeah, that's a great question to start off with, and I guess I would also just say thanks for having me, and what a joy to be able to for us to kind of sit down and have this conversation. I think it'll be really a lot of fun to sort of see where we're going with this program and where ALS development is going. So, people with ALS have a lot of needs. I think if we take a step back, in clinic, we focus a lot on symptomatic needs, on all the things that we can do to help people with ALS. From a field perspective, having more tools to slow this disease would be really, really valuable. And it's not an either or. I think sometimes people sort of create some sort of a false dichotomy to say we either need to focus on care or we need to focus on research to really find a way to slow this disease. And I don't think that there is a dichotomy there at all. We need to do both. We can do both. But there is a huge need to find more targeted therapies to slow the progression of the disease or halt progression, or even begin to, we're now in an era where we can think about, might we be able to reverse some of the progression or prevent the disease altogether? Those are questions we really couldn't have asked many years ago.
Thank you. Well, how has the ALS treatment landscape evolved over the past decade?
I think we've seen, a few developments. A big one has been the approval of tofersen for SOD1 ALS. And this has really laid a pathway for us to say we can have a remarkable effect on the biology of the disease that can have a remarkable effect on slowing, halting, or even reversing some of the weakness that comes with the disease. And we can look for ways to amplify that, but it's sort of a mark in the sand for us to say we can do this well or better, and things we used to wonder whether we might be able to do, we now know we can do. that's for 2% of people with ALS. It's really a very targeted therapy. What we want to do now is replicate that for people with all forms of ALS.
There have been a lot of studies, a lot of potential treatments, a lot of failures, unfortunately, in this area. Why do you think current therapies, those that are currently approved, only show modest benefits?
Yeah, I mean, I think well, why do they only show modest benefits, and why have we had so many, trials that didn't have the positive results that we're looking for? I think many of the things that we have tried for this disease have been somewhat unfocused, and we've tried, study drugs that we thought might have a benefit based on observations we've made or broad observations of neurodegeneration or of ALS. I think we're really entering an era where we're basing our clinical trials now on biology that comes out of the lab, really detailed decades-long work that Dr. Appel has led in his lab really leads us to understand that neuroinflammation is a key piece of what's going on in ALS and specific ways to address it. And I think that's really different today than it was, 10 years ago, where we're really, though we want these things to apply to all people with ALS, we want to be specific in our thinking and in our targeting. And so, they'll say to call Stan Appel a leader in the field of neuroinflammation in ALS is really to belie the fact that he was the only one in the field of neuroinflammation for many, many years. And now I think it's very clear everybody in the field believes that this is playing a key role in the disease that gives us a specific target and a better chance for success.
Great. Dr. Appel, we're going to talk a little bit more in detail about inflammation and oxidative stress and the work that you've done over the decades. But before we get there, would you like to comment on your experiences with patients living with ALS and what the unmet need is?
I'd be honored to do so, and I'm especially honored to have this webinar conjointly with James Barry. I look at him as one of the real leaders in this field and trying to do something to enhance the quality and hopefully even the longevity of patients with ALS, make living part of their looking forward to tomorrow. So the I agree with him in terms of the unmet need. We're getting very, very good at treating symptoms. We know how to treat respiratory dysfunction, and that's made a big difference in our patients. We've been able to enhance their motility. We're emphasizing the problems that may come when you're choking and coughing and can prevent that. We're also knowledgeable about how balance problems can enhance falling, and falling is trauma and can enhance inflammatory responses and slow the recovery of patients. But what James said is absolutely the case. In addition to all these symptomatic, we need better ways to deal with the fundamental biology of disease, better ways to, in the case of the mutant SOD patients, now that we have tofersen, that's very, very exciting, and it's a first. And we need many other firsts like that. But we need ways to target specific biologic processes that are driving and promoting disease, that are enhancing progression and curtailing survival. So and every single patient of ours asks the same question. When are we going to have something that can make a dramatic difference? We've got to go step by step, slow steps, but we're getting there, and that's very exciting.
Okay, well, now that we're talking about targeting specific areas in ALS, let's explore the role of inflammation and oxidative stress in ALS. Dr. Appel, could you elaborate on the scientific understanding of inflammation and oxidative stress as drivers of ALS pathology?
Well, I think all of us know full well that when we're talking about inflammation, we're talking about the immune system and alterations in the immune system that are enhancing inflammation. There are several cells that do it. The macrophages, which are activated, releasing pro-inflammatory, we call them cytokines, that in fact can drive degeneration. There are T cells, whether they're CD4 T cells that are Th1s that can release gamma interferon, or whether it's Th17 that can release interleukin 17, both of which get us into trouble in terms of enhancing degeneration. Or even CD8 cells, the so-called cytotoxic T cells. And these are what's driving the inflammatory process in our patients, as well as in many other diseases, rheumatoid arthritis, etc.
How do regulatory T cells or Tregs fit into this picture? And can you describe the relationship between modulation of Tregs and potential therapeutic approaches?
We're very grateful for the amazing discoveries of Shimon Sakaguchi, because he's the one that first described the fact that Tregs are the neuroprotective cells. They're neuroprotective by their ability to suppress inflammatory responses. And this was an amazing discovery, and colleagues who described the FOXP3 as a pertinent component of the Treg cell aided in that and were also awarded the Nobel Prize. So Tregs are neuroprotective, and under normal circumstances, they'll protect the suppressive and suppress activated macrophages releasing cytokines. They'll suppress the T cells, the T effector cells that could be driving disease, or the CD8 cytotoxic cells, or dendritic cells or natural killer cells. So Tregs are extremely important as neuroprotective agent. But there's a problem. And the problem is in ALS, the Tregs are not suppressive to the extent that they should be. So you have dysfunctional Tregs no longer able to put out the fire of neuroinflammation and neurodegeneration.
So it's clearly a complex and redundant system. And so now let's move towards the rationale for combination therapy, and in particular, the combination of low dose IL-2 and CTLA-4. And for this, we'll start with you, Dr. Appel. Can you describe the rationale for combining low dose IL-2 and CTLA-4 IG as a potential therapy for ALS?
Well, I must say, we didn't understand why IL-2 was not the end-all be-all of solving the whole problem of ALS. It enhances Tregs when you administer it in animals or in people. The problem when you do that is that the Tregs that you make are going into an inflammatory environment. Namely, if you give IL-2 to an ALS patient, we've just talked about how the macrophages are activated, the T cells are activated. And we never understood exactly why, when we were first isolating Tregs from patients, they were dysfunctional. But the minute we took them out of the inflammatory milieu, the interesting thing was they were normal. In fact, they appeared to be super suppressive. So what was going on there? Well, I'm really pleased to say we now have a better understanding of that, which we didn't before. If you take macrophages that are activated and you put them in the laboratory in the company of Tregs, after several days, the activated macrophages turn the Tregs into dysfunctional Tregs. So you take normal Tregs, you take them out, and you incubate them with activated macrophages. It makes the Tregs dysfunctional and exhausted. Well, that's interesting. So how do we deal with that? We deal with that in the laboratory by adding a compound that can suppress some of the inflammatoriness of an activated macrophage. And the one based on the laboratory data that we had that seemed to be safe, available, and could suppress the dysfunction in the activated macrophage was CTLA4 Ig or abatacept. So when we had this in vitro data, we said, gee, if we put the two together, maybe we would prevent the dysfunction and exhaustion of the Tregs by suppressing the inflammatory response. So just to summarize the situation, under normal circumstances, Treg can suppress macrophages. But if the macrophages are very pro-inflammatory with a significant increase in release of cytokines, peroxides, etc., then the Tregs turn dysfunctional. But if we suppress the macrophages with something like abatacept, now we could use the combination because we don't have bad guys turning Tregs into bad guys. They can stay good guys. So the purpose of the combination therapy is for IL-2 to expand the Tregs. At the same time, you're suppressing the inflammatory response of the macrophage, and now the combination is going to be more effective. Well, that was the basis of the trial we did with
Well,
COVID.
before we get to the evidence in the trial that supports the combination,
Sure.
um, Dr. Berry, you've seen a lot of trials. You've led trials. You've studied a lot of mechanisms, so you have a really wide understanding. Uh, what do you think about this mechanism that Dr. Appel described, and in particular, this combination of low-dose IL-2 and CTLA-4?
Well, I mean, I think it's very exciting. I want to sort of highlight that, so Dr. Pell and the people in his lab have for a long time laid out the evidence more and more and more convincingly that regulatory T cells are important when we're understanding the biology of ALS. And there was a time, maybe, gosh, I don't know, almost 15 years ago, I think, where I was working with some colleagues here and looking at monocytes or macrophages, and we're finding this activation of macrophages. And I think one of the things I want people to understand is we often go to these scientific conferences. There are big presentations, but sometimes the moments between these presentations are really, really important. And I remember having these conversations where Stan Appel would come to me and say, "Listen, the Tregs are central to this." I said, "That's very clear from your work, and yet the monocytes look like they're activated too." And he would say, "Yes. Gonna have to figure that out." And I think it's really after putting those two stories together with the Tregs at the center, but the macrophages having this feedback, that it all began to become a little more clear. And the immune system, we can say the immune system is complex. That's an understatement. But one of the things that's very clear about the immune system is that there are a lot of redundant pathways. You said that earlier, Fred, and I think that's true. There are a lot of pathways that have sort of two ways to get to the same endpoint. And if we don't identify both of them and focus on both of them, we may come up short. And I think, we've seen IL-2 have some small benefit on its own, but not in the way that we would have anticipated. And I think it's partly because of this redundant feedback loop that is going on between activated macrophages and Tregs. And so to be able to address both of them with such precision is really exciting.
All right, so we discussed the potential mechanism, the complexity and redundancy of the system, and how the possibility that combining low dose IL-2 and CTLA-4 may suppress that inflammation that leads to the destruction of neurons. What evidence supports use of this combination? Dr. Appel, you were about to discuss experience with this combination in patients.
Yeah, we did a study. It was not a large study, but for us, it was very exciting because it appeared to give us the proof that maybe a combination therapy would be more efficacious than either drug alone. And so we took 4 patients with ALS that were progressing at what we would consider an intermediate rate. Actually, one was a C9orf72 patient, the first patient, and we gave them this combination, and we did it, and we've already published on this, and we did it for a period of the 6 months, and then we extended it beyond. And when we gave the patients this combination uh, every 2 weeks over that period of time, and it was given subcutaneously, both the IL-2 was given subcutaneously and the abatacept or CTLA-4 Ig was given subcutaneously, it seemed to slow progression. What was surprising to us is the suppression in several of them were so exciting that it really seemed to stop progression. But like anything else, if you look very carefully as we continued on, it was clear that we were still making a difference, but it wasn't stopping progression, but it certainly was slowing progression. But the most interesting thing, in addition to how the patients felt tremendously about this, and they asked us to please continue with the therapy, was the biomarkers. So we took biomarkers that we think are meaningful. The biomarker of inflammation, interleukin 18, and we looked at interleukin 18, and in 3 of the 4 patients, we suppressed the interleukin 18 signal in the serum. We looked at oxidative stress. There were 2 of them, 4-hydroxynonenal and oxidized LDL. And in 4 of the 4 patients, the oxidized LDL significantly slowed the increased levels so that it dropped down, and the patient seemed to slow progression in association with that. And we found the same thing with 4-hydroxynonenal, which is a lipid peroxide. So we felt we were accomplishing something. We even added the NFL to that study. Actually, 2 of the patients did not have a high NFL. 2 did have an elevated, and the 2 that were elevated dropped down in terms of the NFL. So in all the signals of the biomarker, not only did we hit target because we enhanced the Treg numbers and suppressive function, but as well it seemed to be slowing or even stopping progression for the 6-month period. So 4 is a small number, but when the 4 seemed to be corroborating what all the in vitro data was saying and all the studies we've done in vivo in the animal models, we're excited that hey, maybe this is the time we need to do a big study with this and move on and see if this can really make a difference.
So it's very interesting because when people hear 4 patients, they say, well, it's only 4 patients. But what you described is that in those 4 patients, over a 6-month period, there was no progression. which was surprising. But even more importantly, as you described, there's biologic correlates that showed and validated the mechanism behind this. And obviously, it was convincing enough for us to now move forward in a controlled phase 2 trial following discussions with the FDA. Dr. Berry, given what was described in those patients, what are your observations? How important are these findings? And what do you think about this data?
Yeah, I mean, I think anytime we come into the question of, should we move forward with the trial, we're going to look at the, sort of the totality of the evidence. There's really, I mean, I said earlier, but decades long evidence that regulatory T cells are important in this process. And I think that monocyte macrophage activation is also important. Um, and then more recently, some emerging evidence about biomarkers that we can look at that might tell the story. So, then we move from kind of the animal models, cell models, animal models, biofluid testing into people. And, sure, 4 people is not that many. It'd be wonderful to have more. So what do you do when you have a good story in preclinical models and a compelling case in a small number of people living with the disease? Is we go on to a larger group of people living with the disease. That's the really exciting thing, is to take promising therapies into larger trials and learn more. The nice thing about this is that the way the 4-person study was designed gave us a paradigm for kind of having a good way forward in a clinical trial. And it, I think, fit into broadly our design for clinical trials, which is that we follow people over a period of time and look at how they're doing clinically, measure biomarkers over that period of time, and see whether we're hitting our target, neuroinflammation, and then see whether that's having an effect on our underlying disease process. Neurofilament light is a biomarker that gives us some window into that. There's still a lot we have to understand about it. I think we can still slow ALS without changing neurofilament light, but if we change neurofilament light, that's important. And then we have clinical outcomes that we can follow as well, and we know that they were useful in the smaller number of patients.
So before we speak and discuss the design of the All-Stars phase 2 trial, there are a couple of other questions that tend to come up. When we use terms like inflammation, people start to think, well, why can't I just lower my whole body inflammation through diet and other kinds of things? Why can't I just take a steroid to lower inflammation? Can you explain, and this is for Dr. Appel first, can you explain why, of course, those methods will not work? And importantly, what is happening at the level of the motor neuron? What is being created that are unfortunately killing these motor neurons when you're in a high inflammatory state?
So, Fred, you've thrown down the gauntlet, because now we get to where not everyone agrees with the fact that the motor neuron injury seems to start distally in the motor neuron outside the blood-brain barrier. But I'm a strong believer in that, and there's a lot of data supporting that. And that the process from the neuromuscular junction back is a dying back phenomenon. One of the things that is really key is a very early event in the injured motor neuron is an impairment of the distal axon and a degeneration of the distal axon, the dying back. And that's associated with denervation. So the nerve is no longer connected to the muscle. Well, how does the muscle and nerve get that neuromuscular junction back? Interestingly, the immune system is key, and we're just doing some experiments that are documenting that. And our friend, the macrophage, is front and center there. So it's not just Schwann cells, terminal Schwann cells. It's not just the presynaptic terminal. It's not just the muscle with the acetylcholine receptors, etc. It's the macrophage that's attracted into the neuromuscular junction, and beautiful data has appeared just recently published on a chemokine receptor sitting on the macrophage called the CCR2 that's attracted to the neuromuscular junction that has been even slightly denervated. Now, this cell goes back into this area, and an elegant experiment came out of Boilet's lab in Paris, who documented, gee, if in the mutant SOD mouse, we take out the macrophages and we put in good macrophages that are not activated, what happens? And what happens is the motor neuron did not undergo degeneration. So here we have a mouse model saying activated macrophages are causing a problem at the neuromuscular junction. And the greater the amount of cytokines released and lipid peroxides released, and lipid peroxides are increasing because of the mitochondrial damage in the presynaptic terminal. All of these things fit together, telling us that's what's targeting the shortened survival and the accelerated progression. So here we have the immune system that's a very important component of the neuromuscular junction and our ability to run, jump, etc., that has been compromised with a subsequent dying back phenomenon. So this is how we think the process is proceeding. And there is no question that the survival, the rate of progression, and burden of disease correlates beautifully with what's going on in the Tregs with their dysfunction, as well as in the activated macrophages.
So once again, by treating the environment and lowering the inflammation and focusing on regulatory T cells, perhaps with this combination, we're really getting at the heart of what could slow or halt this progression. So now let's move Dr. Berry into the phase 2 All-Stars trial, which you're a PI for, principal investigator of that trial. That's a trial that's in close to 25 sites across the United States and Canada. Can you walk us through the design of the All-Star phase 2 trial and what makes it unique?
Yeah. Yeah, no, I think it's a really fit for purpose trial design to answer the questions that are most salient at this point in the clinical development. So I'll just explain that, when we think about how do you take a therapy, in this case, a combination therapy, from not knowing about it in people to, through an approval process if it works. And we all obviously hope that that's where we're going with the development process. We call that the clinical development process. And the first step is to really start in a small group of people, make sure it's safe and tolerated in the population of people that you want to treat, and hopefully get some idea of what you want to measure and what you might see. Usually, we don't get much of an idea of what we might see. With the 4 patients that were treated earlier, we got an idea of what we might see, and that's really exciting. It means we know what to measure as an outcome measure, ALS functional rating scale. It means we have an idea of what biomarkers to look at. And that is not just neurofilament light, which is also 4-HNE, oxidated LDL, and IL-18, as well as a number of other chemokines and cytokines. So that really gave us a lot of information to take the next step in the clinical development program. And each trial is like a stepping stone in the pathway of clinical development. So the next step is the All-Stars trial. And this trial is really designed to bridge the gap from people who say, well, four people, that's just not that much, to knowing a lot about the drug, knowing the dosing regimen of the drug, having a good sense of the effect of the drug, and getting as far as we can down that pathway with one trial. So the goal is to enroll 120 people who are living with the disease. They will be randomized, and there will be a group that's on placebo. That's 1/3 of the participants will be on placebo. and that will go on for 6 months. The other 2/3 will be divided between two different dosing regimens. Everybody will get low-dose IL-2 5 days a week in 2 cycles throughout the month. And one group will get CTLA-4 once a month and the other twice a month. So we have slight differences. Now, one of the really important things about this trial design is that we really anticipate that both of those dosing regimens will work. And we're looking for the edge, which one has an edge. And that may be a tolerability edge, it might be just a practicality edge, or it may be that we have a biomarker signal to say that one of them actually has a more meaningful effect on the biomarkers, maybe even clinical outcomes. But we anticipate both are effective dosing regimens, and we're looking for the very, very best. I think that's really important at this stage in a clinical development program, so we know that we're running with the one that is most likely to be effective in the future. And we don't have to look back after a large trial and say, oh, we didn't really explore, what combinations are really best. It also means that 2/3 of the participants in the trial are exposed to the actual treatment for that randomized control portion. Now that goes on for 6 months. Over those 6 months, we've heard that we expect biological changes very, very rapidly. We think that we could see clinical changes rapidly based on those initial 4 participants, but we also want to hedge against where might we be wrong? What may be that it takes a little longer for it to take effect. And for that reason, and also for the reason of making sure that people participating in the trial get the most benefit, there is a second portion of the trial, which is an extension phase. And I want to be careful about how we talk about it. There's a tendency to call it open label extension. This is actually uniquely designed in that the people who are initially randomized to the monthly abatacept portion of this will go on with that. People who are randomized to the biweekly CTLA-4 will go on with that. The people who originally are randomized to placebo will be re-randomized to one of those other regimens. Now, nobody will know at that end of the placebo-controlled portion what they were getting or what they're going on to get. And that by maintaining that blind, we can continue to kind of use the information, compare these groups. And if we are, if we're not right, if it takes longer to see a clinical effect, we have a longer period of time to compare those and to see what happens when people make the move from placebo treatment to the active treatment or continue on the active treatment.
So as we mentioned before, the primary endpoint is the 6-month endpoint, and that's the one that we'll be moving forward to with respect to our potential application to the FDA, depending on the data. And as you mentioned, the innovative approach of the blinded extension allows us to collect more data, sort of value-added data, if this is an effective treatment, right? Because you would expect that in the placebo group, they would have decompensated somewhat. And we're now starting at a point of decompensation with this therapy to see if it stops as well. And just a final point that I'd like to ask you about this, and that is, it's very important to get patients in that sweet spot where a therapy such as this could have an impact. What are some of the things in this trial are getting to the patient that comes in at a point where you can actually intervene and hopefully change an acute disease into something more chronic, as Dr. Appel mentioned before?
Yeah, no, that's right. And thank you for bringing that up. So the trial's been designed really meticulously to enroll people who are earlier in the disease, so that there are motor neurons that are still available to spare, to help, but also so that the neuroinflammatory process that you've heard Dr. Pell describe so carefully is at a stage where it is most rescuable by this therapy. And so that's defined by the sort of how long people have had disease, what their functional rating scale shows, what their vital capacity is. It's also important in trials to enroll people who have enough pathology, enough of the problem of the disease, that it's causing the disease to change over time. Because That change over time is what we measure to say a treatment is working or it's not working. And if we see people that aren't changing over time because they tend to have a very, very slow progression in and of themselves, then we need to do longer trials. And one of the things we've talked about is we've designed this to be a 6-month trial. In order to do that, we need to have people who are showing some rate of change at the outset. And so we actually have incorporated that into the inclusion criteria for the study to make sure that people who are entering have shown change over time, and they would be the ones that are predicted to change over that 6-month period. That gives us the best chance. So these are things that we do to create less variability in the data over time, which allows us to see the signal better. We think of it as a signal to noise ratio. We're trying to boost our signal to noise ratio. And also in this case, I think, and it's slightly different than some other programs that have taken different approaches, to really get at that sweet spot of not only neurodegeneration, but also neuroinflammation and get our best chance of seeing it. Doesn't mean that this wouldn't work at other stages in the disease, just to be clear. It's just to say this is how we most quickly get to an answer that we can decipher and say, we're sure that this is the actual answer.
Great, thank you. We're at a stage now where we can take questions. There's a lot of questions that actually came through. And so we'd like to go to the question and answer part of this webinar. So the first question is for you, Dr. Appel. How long does it take for observed increases in Tregs and suppression of inflammation with the low-dose IL-2 CTLA-4 combination?
Well, the Tregs in that study, the 4-patient study we did, were significantly elevated with enhanced suppression by week 4. So this is week 4. That is 2 cycles of that, and we saw that difference. In fact, at that time, when we looked at some of the biomarkers, they were decreasing, but it was much more marked if we were out 2 to 4 months where we saw the significant change. The real question behind that question is how long does it take to reverse the process that will allow sustained reinnervation, sprouting of the motor neuron, etc. And that remains to be determined. Hopefully, every time we do a study, we're going to get more information on that. But at least we can say the T cells are going up and stay up after about a month, and the other cells seem to be, namely suppression of the macrophages, seem to be mirroring that, but perhaps at a little slower rate.
So that's interesting because it leads into this second question. Given that mechanism, this question has come up. And this is for you, Dr. Berry. Some studies have extended the primary outcome for the ALS-FRS, as in the All-Stars trial, but they've extended it to 9 months or more. Given what Dr. Appel just mentioned about the mechanism and what you know, what gives you confidence that the Koya study that has a 6-month primary endpoint, even though we know there's also a 6-month extension, is enough time to show efficacy?
Yeah. Well, so one thing I would say is that there are 2 reasons that people have dysfunction of the motor neurons. One is that motor neurons have died, and the other is that motor neurons are under attack and not functioning correctly. And I think it's not unreasonable, given what we saw in those initial 4 patients that were treated, with a fairly rapid clinical response or maintaining of clinical function, that by reducing the neuroinflammatory assault, we may actually normalize some of the functioning of the cells that have been under attack and not functioning properly, but not lost. And so I do think that if we can, as you just heard from Dr. Appel, I think we can think that if we halt neuroinflammation, there will be sprouting and there will be, recovery of innervation. But I also think before that, there's some substantial chance that we may see improved function of the existing motor neurons. And so that could be a reason that we saw and could see function that returns faster. I think, you know, we need to find a balance between looking at the biological impact of the drug and looking at the clinical impact of the drug and giving ourselves that additional kind of potential benefit of, gee, I'm not right every time. I think 6 months is enough time, but we do have additional follow-up in case we need that to supplement what we see as the primary endpoint.
Hey, this next question is about safety. We've spoken about mechanism, we've spoken about potential efficacy. Uh, what is the safety profile from what we know now of this combination and of Koya 302? And before you answer, I should just note that Koya has done a large toxicology study, preclinical toxicology set of studies with the combination and have not found any significant toxicity. But perhaps you can comment on what you know of the data from humans from a tolerability and safety profile, Dr. Appel, since you conducted it at Methodist.
Yeah, I think the one thing that I can say that I'm absolutely certain about. Now, there aren't many things that I say I'm absolutely certain about, but I am here. IL-2 has been very, very safe over many studies, over many years, Europe, United States, what have you. And our study really proved to be the same. People worried that maybe you're enhancing infection, the likelihood of infection, because what you're dealing with is you're dealing with something that suppresses the macrophage's ability to fight off bugs, etc. We never saw any problem with respect to an increased incidence of infections or any untoward response. And so I'm very comfortable that with respect to safety and tolerability, we're going to pass that one with flying colors.
Well, the other thing to note is that this is a subcutaneous formulation. So this can be administered either by the patient or caregiver. Dr. Berry,
Yeah.
you going to say something?
Yeah, well, I wanted to jump in with a couple of things. So first of all, that's right. The subcutaneous injection is just like insulin, goes in that same space that insulin goes in. It's not as deep as a vaccine goes into the muscle. It's just under the skin, so quite easy to do. Um, and there can be some redness at the site of injection of IL-2. I want to point out that we're using IL-2 at a very low dose, and one of the reasons that works is that IL-2 actually activates T cells. And so at high doses, it activates all those sort of, pro-inflammatory T cells. But at these low doses, it preferentially activates just the regulatory T cells. So its effects at these very low doses that we're using are very different than at super high doses where it can cause, more pro-inflammatory response. We don't see those at these low doses. We've used it in trials together. Dr. Pell and I, it's been used in Europe in large trials. It's been used off-label. I agree that it's very, very safe. A beta-sept, CTLA-4, is also, it's broadly used at these doses. It's used for inflammatory diseases to reduce the inflammation. It's used quite effectively. It's used quite safely. And so there's also a lot of experience with it. One of the benefits of using a drug that has an indication is that there's a lot more information about it. And so, we understand it really quite well. And we do need to think about making sure that people don't have latent TB infections. But the point is we know to look for those things, and that's built into the trial and would be built into any use of it after that.
Okay, I think we have about 10 more minutes here, and we have more questions than we can get to, but we have three more questions that I'd like to get to. The first one is, if Koya meets its primary efficacy endpoint after 6 months, do you expect prolonged survival during the 6-month extension and beyond? Let's start with Dr. Berry and then Dr. Appel.
Look, I think if we can, there is a very close correlation between the slope of the ALS-FRSR and survival. That's true in natural history studies. That's something that we see. It's not a perfect correlation, but it is a close correlation. And so I think if we can slow disease, but over a shorter period of time, looking at the ALS-FRSR, that sets us up to look for long-term survival as the next thing that we would see a benefit in. This has been true in tofersen, clearly true. Um, and so I think we would be very, very bullish on the idea that we could look next at survival and see a benefit in survival if we see a benefit in ALS-FRSR slope.
Dr. Appel, do you want to comment on that?
Yeah, I totally agree with James, but uh, let me play the cynic, which is not usually my role. The question is, if inflammation is enhanced despite our efforts, then my concern would be that there might be slowing of the suppression of the inflammatory response. I'm not saying that will happen, but that's something we're going to want to monitor very carefully in the next 6 months or beyond that. There is no question in this period of time that, as James has said, if in fact you're suppressing inflammation during that 6 month period to the point where the ALS FRS is not dropping at the rate that it did prior to initiation of the study, then we'd be okay. Namely, we're still going to have a safe and tolerable and something that if it slows for 6 months, it must have an effect on survival. The progression and survival are intimately linked.
Okay. The next two questions I will answer. The first one is, does Koya plan to have an extended access protocol or compassionate use program? And the answer is yes. We are in discussions right now to create that extended access protocol for patients that might qualify. And the last question is, does Koya expect approval by the FDA if this trial meets its primary endpoint? Well, this is a review issue by the FDA. They will review the data. They have approved therapies before on a single trial, and it all will really depend on the strength of the data. So if the data are strong and we are in fact slowing or halting the progression, then we will submit this application for approval. So that was the last question, and I would like to thank our panelists and those listening to this webinar. I hope the webinar was informative. And as a reminder, to learn more about Koya's ongoing All-Stars trial and participating sites, please go to trials.koyatherapeutics.com. I'd like to thank our panelists, and I'd like to thank those who are listening, have listened to this webinar. Thank you.
Thank you for having us.
Thank you for having us.